Hi, everyone. Thanks for coming. This is our graduate invited speaker, Dr. Andrew Nuss from University of Nevada, Reno. He's in the Agriculture, Veterinary, and Rangeland Science Department. He's not unfamiliar with Purdue. He did his master's and his bachelor's degree here back in like 2000. Went over to University of Georgia for his PhD. But because Purdue black hole and will always suck you back in, he came back for a postdoc in 2013. And that's where we overlapped in Dr. Hill's lab. And today he's going to be talking about insect olfaction and some neuropeptides from bloodsuckers to sapsuckers. So it'll be hopefully pretty broad and interesting.
All right. Yeah, thanks for that introduction. As noted, I've kind of been to Purdue and left and come back, and as an entomologist I start to get some insect metaphors in my mind, and it's like, you know, moths circling a flame, which usually doesn't work out too well for the moths, so maybe I'll just put that aside. But it's great to be back here, and really I'm very appreciative that the grad students selected me to give this seminar, so I hope I do it justice. But hey, it's wonderful to be here. Purdue's a great place, and it's good to see familiar faces, and I want to meet a lot of people that I haven't met before too, at least the faces that I can see. I think we all thought we'd be out of this pandemic by now, but anyway, so yeah, as you might know from my title, I'm going to talk about two pretty different areas of my research program. There's certainly a lot of things that I'm working on and I'm excited about, but I'm just going to start out with our work, our DARPA-funded project on mosquito odor receptors. So as you may be aware, mosquitoes and other arthropods are vectors of numerous human diseases, and they're responsible for millions of deaths or cases of morbidity annually around the world. And since the discovery that mosquitoes can transmit disease, there have been a lot of efforts in managing pest populations to try to prevent the spread of these diseases. One of the more prominent techniques is the use of chemical insecticides, and the idea is pretty obvious, right? You know, dead mosquitoes aren't going to transmit disease. So that's worked out okay for a while, but there are some drawbacks to these lethal techniques. You know, one is the development of resistance. Even this, you know, little demonstration I did slapping my hand. You know, mosquitoes, some species feed on us at night to avoid being killed, and certainly there are strategies for development of chemical resistance as well. Multiple types of strategies for the insect. Another disadvantage to using lethal techniques is that if you do ever relax the control efforts, there are a number of reasons that might happen. Mosquito populations can rebound rather quickly. So there are about 3,500 mosquito species in the world, but as far as human disease is concerned, there are only about 200 to 300 species that are really of deep concern to us. And the ones that are the worst for humans are the ones that specifically target humans and more rapidly perpetuate the cycle of disease transmission from human to mosquito and human again. But if you think about it, it's not too hard to see why. There might be some human specialist mosquitoes as we've gone from villages to towns to cities, where this large animal that's a rather stationary food source. So some mosquitoes have adapted to that situation, but there are a number of other species that prefer, are more generalist or prefer other animals, reptiles, birds, cattle, and so on. The way that they're able to differentiate humans from other animals is that we emit different odors from our skin microbiome, also our breath. And these mosquitoes have different odor receptors that are more or less receptive to certain chemicals that are emitted by their preferred hosts. So the study of mosquito olfaction is a pretty competitive area. There are a lot of groups looking for repellents, of course, for obvious reasons. There are also groups that are looking for attractants with the idea of attracting mosquitoes to something like an insecticidal bait to, again, limit populations. But there are two advances in biology that I just want to highlight, and they've certainly advanced other areas of biology, but for mosquito olfaction in particular, I want to mention high throughput sequencing. Certainly we've had a genome for Anopheles gambi for about 20 years now almost. But getting other mosquito species genome sequence has taken a little while, but once that cost of sequencing has gone down, the proliferation of genomes available has expanded greatly. So I have 17 Anopheles genomes here on this slide. This is a bit dated. There's certainly a lot more available. But there was one paper in particular, Nifsi et al. 2015, that I wanted to highlight, where they did 16 Anopheles genomes. So Anopheles is the genus of mosquitoes that's responsible for human malaria transmission. So this was notable for a number of reasons, and they sequenced a number of vector species, shown here with squares or circles here. But also interesting in this study were these little triangles here, and these are non-vector species. So this gives the possibility of starting to look at this question of how do some of these mosquitoes specialize on humans versus other animals. Quadria annulatus in particular is one that's part of this Anopheles gambi complex, but it specializes on cattle. It prefers cattle, I should say. It's a competent malaria vector, but it just doesn't feed on people to pick up the malaria parasite in the first place. Okay, so we can make some of these comparisons, and another huge advancement was CRISPR-Cas9. If you don't know what that is, look it up. It's pretty cool. But simply put, it enables genome editing, and especially in non-model organisms. So we previously had some tools to modify mosquitoes, but they were very challenging to use and imprecise. But this gives us much more precision in the process. So, with that kind of background and these tools available, we had this idea of, could we figure out which receptors are most responsible for selecting human hosts in these most deadly species? And we have a couple approaches for looking at that. And that's mostly what I'll talk about today. But if that's possible, can we disable these receptors or modify them or replace them with receptors from other species that prefer other animals, and modify what host they're selecting in the environment and drive them away from humans towards cattle or other animals in the environment? And the idea generally, essentially, is, can we create a human-avoiding mosquito? So if this is possible, we may be able to drive this trait into the population using gene drive mechanisms. Again, I'm not going to talk about gene drive today, but there are a number of groups working on that for various reasons, usually to crash populations, but in this case, it would be more of a population replacement. And if this is possible, you may be able to break that disease transmission cycle. If the mosquitoes are feeding on other animals, not feeding on people, they're not going to pick up the parasite in the first place and transmit these diseases. Some other advantages to this technique is that we're avoiding some of those downfalls of lethal strategies. So, we allow the mosquitoes to still survive. They can feed on blood, just don't feed on humans. So there's less selective pressure on them. Also, this may be something that can be retained in the environment, even if the management efforts are ever relaxed in any way. If you have some sort of disruption, again, political instability or lack of funding, that mosquito population may be able to maintain itself with that gene drive mechanism to prevent reinvasion of human-preferring strains. So I know there are a lot of ethical issues and public perception problems with introducing genetically modified organisms into the environment, but we may be able to convince people mosquitoes that don't bite humans are a good thing. So, okay. So we chose to focus on Anopheles stevensii. This is more commonly referred to as the South Asian malaria mosquito because it occurs in Southern Asia from the Middle East all the way to China, and it's an important malaria vector in those regions. But it could also be termed the urban malaria mosquito because unlike a lot of Anopheles, this particular mosquito likes to, it is capable of laying its eggs in small containers, artificial containers like cisterns, things that occur in cities. So it's able to survive there. And it also is a pretty good biter of humans. So we thought this was a good candidate to approach this insect, and I dealt with it in the past as well. Okay, so how do we get started on a problem like this? Well, we started with looking at Anopheles gambiae. This is the most probably best characterized mosquito that transmits malaria. I mentioned we had a genome for many years, and it's the best characterized genetically. It's also an infamous vector of malaria in sub-Saharan Africa. So it's been heavily studied. And in that species, there are 71 odorant receptors. I realize I didn't provide too much of a background of chemoreceptors in mosquitoes, but I'll be talking about odorant receptors today. And in Stephen's eye, with the genomes that we had available, there were 58 odorant receptors that we saw. Now, there may be some lurking out there. I'm not totally sure that these genomes were as complete as they could be for Stephen's eye, but this is the landscape that we were working with. So we also had to get a starting point for where we were going to, which receptors we were going to look at. And others have approached this kind of idea as well, or at least which odorant receptors are important for host seeking, maybe not specifically human host seeking. So one of the ways this has been approached was by using transcriptomics. So the idea is to take host seeking females in species that are human host seeking and compare these transcriptomes from the antennae or mouth parts to mosquitoes that are blood fed, with the idea that once they've taken a blood meal, they're not interested in looking for host, so they may be downregulating these odorant receptors that are important specifically for host searching. Also, the males don't take a blood meal, they don't host seek in these species. So again, they shouldn't theoretically be expressing these odorant receptors that will lead them to host. Also, there have been comparisons between closely related species that are either human preferring or prefer other animals. They're zoophilic. So I mentioned Quadria annulatus, for instance, prefers cattle. It's very closely related to other species in that Anopheles gambiae complex. So comparisons between these species are also pretty interesting for that question that I raised. And through a synthesis of the literature and these types of studies, we've identified about 10 receptors highlighted here in red that we think may be involved in human host selection. And conversely, there were some receptors in blue there that were upregulated in the zoophilic species. So we may have something that kind of modulates this shift, possibly. Okay, I'm going to talk about these others here in a minute, why we picked those. But first, we had to address kind of the elephant in the room is Anopheles gambiae and Stephen's eye are separated by about 30 to 50 million years of evolution. That's kind of a long time before humans were even on the scene officially, and certainly before we started making towns and cities. So one of the things we thought about is that they may be coming about human host selection from different angles. It may be a question of convergent evolution, in other words. So they may have co-opted different odorant receptors to adapt to the human odor profile. So one of the things we wanted to check was first, does the antennal transcriptome of Anopheles Stephen's eye look like the profile in some of these other species? And also we wanted to look at the mouth parts. These are important sensory organs as well. So we selected RNA antennae from non-fed females, females that had been blood-fed, as well as males, to do some of these comparisons that I was talking about earlier. These were insects that were dissected in the evening. That's when they should be at their peak of host seeking. And this was quite an undertaking. This is pre-COVID, by the way. That's why we're not wearing masks, but remember that. Yeah, we had to get the whole lab going. Everybody in my lab contributed to this because it turns out there's not a lot of RNA in a mosquito antenna. And after a couple failures, we really ramped up things. It took about 3,000 antennae, about 1,500 mouth parts. There were a couple of undergrads that were particularly good at, plucking these antennae. I think they're going to have good futures as eyebrow shapers if that's where they decide to go. But yeah, this is a big undertaking, but we finally got enough RNA to get this to work. So I'm going to show you some results. I don't want to dwell on it too much. And again, we're only focusing on the odor receptors here. There are a lot of other chemoreceptors I can talk about too. It's a pretty complicated situation. And here I'm only showing the non-fed females here. We didn't see a lot of differences between host-seeking females in males or host-seeking females in blood-fed females, which is kind of interesting too. I mean, and it may be just that there's a basic complement of receptors that are expressed, and the insect decides what to do with them depending on the situation. But I'll mention that a little bit more in a minute. But this does give us a profile, at least for the species. And we can compare to odor receptor expression in other species where this kind of thing has been done. So we have this Athri et al. Paper that looked at Anopheles kalusii and quadrannulatus. Here et al., a little bit older study, compared between Gambi and quadrannulatus, but you can see even these two studies don't really agree completely, and there may have been some differences in study condition. I think Athri et al. Was a little more careful about collecting their antennae in the evening when the insect should be host seeking, but I think there's probably some other things going on there. And Pitts et al. Compared female antennae to male antennae. And there are other comparisons that have been done too. As I mentioned, blood-fed mosquitoes versus ones that are still host seeking. And I didn't put it on this slide, I'd be a little too busy, but we do see some patterns that start to emerge. And what might be going on in Stevens eye versus some of these other species. And one thing I'd like to point out is we've got photoreceptor 11 and 76. Okay, they're pretty highly expressed in Stevens eye as well. But there are some other receptors like OR15 and OR55 that are not present in Stevens eye. Oh, that's kind of interesting. Maybe they didn't get picked up in the genome because I mentioned may not be as complete as we'd hoped, but they may truly be gone, and it may be a result of gene duplication in the arm that went to the Gambi complex, or they may have been lost in Stevens eye. So we wondered, are there other receptors that are kind of picking up the slack? And kind of interesting, OR24 and 48 are pretty highly expressed in Stevens eye compared to some of these other species. So they may be picking up some functions that these other odorant receptors are doing in other mosquito species. Before I talk about the individual odorant receptors, I want to talk about the mouth parts too. The expression profile was very simplified in comparison. We saw OR8 and OR28 was very strongly expressed. And this is something that shows up in other species as well. The picture, at least I get from this, is that the mouth parts are more broadly tuned towards just general odors any host might emit, and the antennae are a little bit more discriminating, because in the mouth parts is also where you'll find the carbon dioxide receptors that are expressed. There are some of the gustatory receptors. There are 3 of them that make a complex, but yeah, it may be a bit more broadly tuned. But as I'll show you in a bit, these receptors also may have some more fine tuning to them. So and that's a good transition to talking about how we look at the odorant receptors themselves, because transcriptomics tells us something, but not everything is necessarily under transcriptional control. Or it might be advantageous for these mosquitoes to be expressing all the receptors all the time. I just mentioned carbon dioxide. They use that in host seeking, of course, but when the insect's blood fed, do they need to sense carbon dioxide? Well, maybe. I mean, there may be other cues from the environment. Certainly, high carbon dioxide concentrations are toxic, so the mosquito might want to avoid that. So they just may be getting that information and not always using it, or using it in different ways. So one way we can look at individual odorant receptors is using this Drosophila empty neuron system. And I'm not going to get into too much explaining how it works, but just briefly, what we can do is insert a odorant receptor of our choice into the Drosophila genome so that it expresses in specific sensilla in the antennae, where the endogenous receptor from Drosophila has been deleted. So that's where you get the empty neuron that this receptor is being expressed in. Then you can take a microelectrode, place it on that sensillum, and puff different odors onto the antennae. And depending on whether it's excitatory or not, you can get an idea of what odors activate this odorant receptor. So this has been used for a number of different insects, so it is a pretty well characterized system. It's not something we came up with, but it's been used to characterize other mosquito receptors as well. And this is actually how we came about looking at some other targets, because in again, in Anopheles gambi, some of these receptors that were characterized are responsive to odors that are found in the human odor profile. So we're interested to see if Stevens eye also uses these receptors. And I've highlighted OR8 because that's all time to talk about today. But again, that was part of that mouth part transcriptome. It was highly expressed. Okay, just a quick slide here talking about the odors we selected. There are thousands, if not millions, of odors we could potentially test against these receptors. But obviously, we have limitations of time and money. So we trimmed down our panel to a realistic amount of about 50 odors we could test on these receptors. And we had a preference for choosing chemicals that were found in the human volatile odor profile, either from breath or from skin. We tried to select some animal odors as well. There aren't a lot of GCMS profiles available from cows. People don't want to know how cows smell, I guess, but we did find a few, and a few from mice. So this is certainly an area that could be expanded. But anyway, we did have a panel to at least give us an idea of what might be going on, what these receptors respond to. So I'm going to show you some of the typical data here. Again, this is OR8, one of these receptors of interest, and these are some of the top chemicals. Obviously, we tested all 50. Paraffin oil is our control, shown here on the bottom. You can see it doesn't elicit very much response, but some of these odors are pretty excitatory when that receptor is expressed in the soft antennae. Another thing I want to note from these top odors that elicited a response, I've got an asterisk by all of these chemicals that are found in the human odor profile. Quite a few. So this might be a good receptor for one of our targets to start modifying. But also, this comparison between species was really fascinating to us. So here's the receptor in Stevens eye on top, aligned with the receptor from Anopheles gambi on the bottom. You can see there's a lot of overlap. These receptors haven't diverged too much, but there are some small changes. And we also have a characterization profile from previous work done by another lab. So this Carey et al. Paper is shown here on the y-axis, and then our work in Stevens eye is down here on the x-axis. And so, for some similar odors, if you think if these receptors are responding exactly the same, we kind of see a nice diagonal line. And for some of these odors, that is the case. For some, they elicit a stronger response than the gambi version of the receptor. But there was one 6-methyl-5-heptene-2-one, also known as sulcatone, that activated the Stevens eye receptor, but not the gambi receptor. And this particular compound is really interesting to us because of some previous work that was done by the Leslie Vauxhall's group. And this was a really elegant study where they were looking at Aedes aegypti. There were these two strains that, same species, but there was a forest strain, and which was more of a generalist mosquito. And there's what they call the domestic strain that survived in the cities that preferred humans. And they looked at a lot of differences between the genomes of these insects, and found out that there was an odor receptor 4 that had an increased activity when sensitivity to sulcatone, which is something that we emit a little bit more abundantly than other animals. And they suspected that this was driving the more anthropophilic feeding behavior of these insects that were found in the cities. So that's kind of interesting in light of the human host preference. Now, I don't want to imply that gambi can't sense sulcatone. This study Carey et al. Characterized a number of other odor receptors in gambi, and there are other ones that can sense sulcatone, but it kind of goes back to that what I was mentioning about that they may have come to human host specialization by co-opting different odor receptors. And more to that point, there's another study I'd like to highlight, Hughes et al. In 2014. There are two similar receptors, OR13 and 15. One is more responsive to acetophone, the other one is more responsive to 4-methylphenol. And what they did was start substituting amino acids out, making the receptor more like the other, and figured out at what point this receptor started switching more to the preference for the other odor. And the key amino acids were highlighted here in green and red. So we see in the second extracellular loop and the third extracellular loop Some in the transmembrane region as well were important for this kind of the odor specificity. And parallel studies in Drosophila have kind of confirmed this, that the carboxy terminal of the molecule is kind of the business end, at least as far as odor discrimination for odorant receptors. So going back to our alignment between Stevens eye and Gamby, if we look at these transmembrane domain regions, 5, 6, and 7, they're identical. So probably that's not changing things. But when we go to the second extracellular loop, we see 4 amino acid changes here that may be driving, and some are not conservative substitutions. So this could be the mechanism that is changing slightly odor preference. And we haven't confirmed this yet. That's something on our list to go to next. But I'm going to kind of wrap up this part of the talk and just mention we haven't modified mosquitoes yet, but we do have some targets to approach this. And once we do get to this point, we're going to be doing some olfactometry. Many of you recognize the Y-tube olfactometer, where we pipe down different odors, and the mosquito can fly upwind and make a choice. Of course, we're trying to get them to go to the other animals in the environment. And as I mentioned before, the long-term goal is to develop something that we can put in the field. But there's a lot of things that need to go right before that happens. So, but it's promising, and it's another way of looking at a problem that we've just been trying to hit on the head chemically with this hammer for years and years. And we've been spraying chemicals since at least the 1940s, and we still have mosquitoes and we still have vector-borne disease. So maybe the best strategy is living alongside mosquitoes instead of killing them. So, okay, so with that, I'm going to shift gears and talk about neuropeptides and Lagis hesperus. So I had studied neuropeptides for my PhD and for part of my one of my postdocs, and so this is an area I was really interested to incorporate into my new lab when I started in Nevada. But I also wanted to make a contribution to Nevada agriculture, because I am in the Department of Agriculture along with veterinary and rangeland scientists. So Lagis hesperus really fit this bill pretty well. And I was able to kind of put these two interests together with the help of Drs. Joe Hall and Colin Brent at the USDA facility in Maricopa, Arizona. And I'm going to take a minute to introduce Lagis hesperus. I think there are probably a few Lagis enthusiasts in the audience. I think I met somebody yesterday that talked about that. So, but anyway, this is a good pest for Nevada agriculture, because this is something that prefers alfalfa. That's important in the state for seed growth, for producing seed, but also for feeding cows. That's our biggest agricultural output in Nevada. But economically, Lagis hesperus causes more dollar damage to strawberry and cotton crops. In California alone, causes about $70 million in damage annually to those two crops. And that number's probably gone up since I made this slide even. But they are able to feed on a number of other host plants. This is a really adaptable insect. Maybe they won't prefer those plants necessarily, but they can feed on them. And a number of these include crop plants as well, like celery. And the nature of their damage is not so much that they just gobble up the cropness, they more feed on the developing fruit and cause like strawberries to form these kind of cat face strawberries is what they call them. Sure, they're fine in smoothies and whatnot, but we got to have perfect fruit in grocery stores. So there's a cosmetic issue as well. And I want to go a little bit further and say that these insects are really true omnivores in many ways. They can feed on other insects as well, to the point that we have to put these kind of protein diet packets in our colonies, or else they'll cannibalize each other. So sorry if the sapsucker part drew you into this talk. I was a little misleading about that. If you want to storm out, it's okay. But yeah, these are more, they'll feed on a wide variety of things. And as I mentioned, pretty adaptable to the point where they have become resistant to a number of major insecticides as well. So they've certainly earned the ire of growers to the point where molecular resources have been devoted to characterizing these insects and trying to find new ways to manage them. And that's how we got to a transcriptome for this insect, in large part to the efforts of my collaborators. So from this transcriptome, there are a number of neuropeptides that have been identified. And we're seeing most of the major groups we'd expect to see in other insects. No major deletions or overduplications. But that kind of raises the question that what's been characterized in other insects, does that apply to Lagus hesperus? And the canonical laboratory insect, Drosophila melanogaster, okay, there's been a lot that's been learned from that because of all the genetic tools available, but do all lessons that apply in Drosophila apply to Lagus hesperus? Because these are pretty different insect groups. They're separated by an estimated 350 million years. That's Carboniferous, an era before the dinosaurs. Kind of a lot's happened since then. So maybe some lessons apply, maybe some not. A bit about insulin. I know many of you are probably familiar that insulin does some important things in us as well as other organisms, mainly regulates blood sugar levels. And that's true of insects as well, but insulin does have other functions. And there are also other peptides that have similar structure to insulin that have other functions. So there's like insulin-like growth factor, for instance, which is well named. It's a growth factor. Relaxin also occurs in our bodies, has important roles during pregnancy. And insects, as well as other invertebrates, have insulin-like peptides, which I've abbreviated here as ILPs. But there are some shared functions, also have important roles in carbohydrate storage, reducing circulating sugars. But there are also roles in growth and aging, some specific to insects. Mosquitoes, there are roles in blood meal digestion, reproduction, immunity, diapause, the list goes on and on. Some according to certain insect groups. But there's been less studies done on hemipterans. Some of those lessons still seem to hold true, but there's a lot that's not been explored. Some indications that carbohydrate storage is also managed by insulin signaling, also wing length. So there may be some growth factors hidden in there as well. So moving more, I always skip ahead on this. I wanted to talk about the structure of insulin really quick. This is normally synthesized as a single chain molecule. Of course, we have the amino acids listed here. And disulfide bonds form between these key cysteine residues. And afterwards, there are dibasic cleavage sites that cleave out this C-peptide here, leaving a two-chain molecule. That's the very typical insulin structure. Insulin-like growth factor retains this C-peptide. It has a kind of truncated C-peptide and doesn't have these dibasic cleavage sites. On the next slide, I'll talk about that a little bit more. So we have found 4 insulin-like peptide sequences in Lagus hesperus. I'm not going to talk about this 4th one today, but for these other 3, they do have characteristics that are the hallmarks of insulin signaling molecules in other organisms. So we have these very conserved cysteines, very consistent spacing for forming those disulfide bonds. Also, these dibasic cleavage sites for getting rid of that C-peptide. So that's for the B and A chains. So one thing I do want to mention, though, is this ILP3 is a little bit atypical compared to those first 2 molecules in that it has a very truncated C-peptide, kind of similar to those growth factors that I was mentioning earlier. Some people have noticed this in other insects and have termed them arthropod arthropod-like insulin-like growth factors, which is a ridiculously long name. And I don't think it's very accurate either because, just hanging the name growth factor on it doesn't, I think there are more functions, which is I'll talk about in a minute. But this is fairly typical of what's seen in Hemiptera, this kind of reduced number of insulin-like peptides. In other insect groups, flies may have from 5 to 8. Lepidopterans have about 30. I didn't want to jump into such a complicated system. So this is a nice system to get started on and start picking apart. So where are these expressed? We looked at several different tissues, the head, digestive tract, reproductive tissue, as well as the thorax and abdominal wall in females and males for ILPs 1, 2, and 3. I think you can interpret this fairly easily. Mostly saw expression in the head. Again, that's not too atypical. We have our pancreas, right? But insects kind of have their pancreas in the head, in their brain. is where they release their insulin-like peptides. So we've got a lot of functions we want to explore, but we thought we'd start with the easy stuff, kind of the slam dunk. Can the insulin-like peptides and ligase hesperase modulate circulating carbohydrate levels? Can they regulate blood sugar in this insect? So we came up with this assay that's very simple. It's like an insulin stress test. Like, has anybody ever had to do that? Like a doctor's office, like you drink a big thing full of like super sugary substance, and a couple hours later, they take your they take a blood sample, and if insulin's working properly in your body, you're able to reduce those glucose levels. If it's not, then there may be a problem. So we did the similar thing with insects, except we injected them with trehalose. That's typically the circulating sugar found in insects. And then harvested them a few hours later and looked at, used a trehalose assay using anthrone to monitor those carbohydrate levels. So first, we have insects that are just taken directly from our colony, no injection or anything. This is kind of where we assume the baseline is. This is resting trehalose levels. Of course, after we inject the insects and harvest them immediately, they haven't had any time to get rid of that trehalose. So this is kind of the high water mark up here. But after 6 hours, they're able to reduce those trehalose levels back down to normal circulating sugars. Okay, so I mentioned those ILPs were expressed in the head. So what we did was take a human hair and ligate the neck of the insect so that they can't release those factors, injected them with trehalose. Even after 6 hours, they're not able to get those sugars out of circulation. So, that tells us at least it is some factor in the head that's being released, but how do we know it's insulin related? So one of the things we did was ligate insects again, inject them with trehalose, but co-inject them with human insulin. So we weren't able to synthesize the ligase insulin. I'd like to do that someday, but it's a little more complicated because of all those disulfide bridges we need to form and everything. But with a dose of human insulin, it was able to reduce things back down after 6 hours to resting trehalose levels. So it really suggests that insulin-like peptides are responsible. But which ones? So we can use RNAi to explore this question. So we knock down each one of these individual ILPs. We also have this, what we term Venus. It's a yellow fluorescent protein. It's just our RNAi control. So it shouldn't be impacting the insect. But again, this is all before we inject trehalose. So we have the colony baseline levels, and the other ILPs are kind of hovering around that resting level. But again, when we inject the insects with trehalose and harvest them immediately, again, that's going to be the highest it's going to get. But after about 6 hours in our control insects, they're able to reduce those levels. Maybe not completely down to the colony level, but there's a huge reduction in the circulating trehalose. But you can see with ILP 1 and 3, they're having a problem getting rid of that trehalose. They're not retaining all of it. Some of it is disappearing from circulation, but a lot of it's still there. So that's kind of interesting because, we expected maybe there was the one insulin that did everything. Possibly there's some redundancy in function. But another thing we were thinking about is, maybe expression of one is dependent on, release of one is dependent on release of the other. They might be co-modulating each other in a certain way. So the next obvious step is what happens when we knock out both of them or all of the ILPs. So we did this, and I'm not going to present the trehalose data today. We're still kind of finishing up some stuff with that, but I do want to note a kind of side effect of trying to knock down these ILPs in combination is that it was pretty darn lethal, especially these this combination of ILP 1 and 3. About 90% of the insects die before we can finish the experiment, which complicates things a little bit. Come on, somebody live. So that was interesting. But what was even more curious is it's not just the knockdown of those ILPs that's causing a problem, because when we knock down all 3 ILPs, 1, 2, and 3, survival is still not great, but it's a lot better. So that's kind of weird. It's not always what we're taking away, but what we're allowing to stay there. So in this situation, the ILP2 is being expressed kind of unfettered. So why is that so particularly lethal to the insect? We don't have an answer to that yet, but we are exploring that question. So this was a little bit unexpected. So the the peptides, the signal themselves are one part of this equation, but the other side of it is what receives that signal. Of course, you have to have a receptor to pick up what these hormones are doing in the system. So we have a couple of candidates for that. Their receptor tyrosine kinases are typically the insulin receptor that we have and many other insects do. So there are a couple of candidates that resemble those in other insects that we're looking at. There are also leucine-rich repeat GPCRs. This would be in humans, we have the relaxin receptor that is a GPCR. That's kind of similar to this. So there is some similarity in that respect. And we do have a couple of candidates that might be responding to some of these insulin-like molecules. And it's kind of cool that we have 4 potential hormones, 4 potential receptors. Maybe there's a one-to-one match. I've had so many disappointments in research projects that I can't believe it would be this simple. So we'll see, but it is a little bit tantalizing. And I know we're probably a little short on time. Maybe I talked a little fast, but I do want to just briefly mention about the receptors. Again, I'm not showing the Trailers data today, but one thing we've started using is RNAi, but another tool that's available is using insulin signaling pathway inhibitors. So there are a lot of drugs that target parts of our insulin signaling to treat diabetes or whatnot, or just to understand how the systems work. So there's a lot of pharmacological tools available, and we've started applying these to the insects. I was a little bit coy about putting the actual names on here. This is pretty early on. I don't expect you're going to steal this, but anyway, some of these are inhibitors of the insulin receptor itself. Some inhibit the insulin-like growth factor receptor, and other parts of the cascade. There's a downstream from that. There's a MAP kinase pathway. FOXO is a downstream target of that as well. And there are a couple others that just aren't on this slide. But again, just looking at the mortality or morbidity of the insects, because we do these assays a little bit differently. The RNAi, we inject it, wait 3 days for it to take effect. But with these inhibitors, we can do it immediately. We can co-inject with the Trailers. But even when we do that, we get pretty high mortality. So this DMSO is our control, getting 0% mortality, but some of these inhibitors cause as much damage as what we're seeing with that knockdown of that combination of ILP1 and 3. So it's kind of an interesting parallel, but this does suggest we may have some tools to start manipulating the system. Maybe we need to back off on the dose a little bit, but we may be able to interfere with insulin signaling so we can start picking apart this system to some degree. Also interesting from an applied standpoint, I worked with Dr. Hill when I was here, and we were looking for analogs to interfere with insect receptors, a different class of receptors, but this is something that could be put along the same pathway. Can we find ways to interfere with insulin signaling in insects to cause them to die? I mentioned drawbacks of chemical strategies, but hey, that's the world we live in right now, and they are important tools. So that may be a direction to follow up on. Okay, so yeah, starting out with this, looking at the carbohydrate storage is again, kind of the easiest way we can get into this system, but there are a lot of other physiological aspects we want to explore. The insulin signaling has a lot of other roles, may have a role in diapause. Reproduction is another target. We're really looking at growth factors I mentioned. Insulin signaling has a role in aging. Could we get these insects to survive a shorter amount of time in the field? That might be a way to manage them as well. So there are a lot of different aspects. We're bursting with questions and are developing some of these tools to get things going. It's just a fun system to work in. So with that, I'd like to thank a lot of people that made this work possible, and funding agencies as well. I want to highlight Zachary Speth here in particular. He's worked with the mosquito odor receptor project, and Devin Mazalewski has been working on the Lygus bugs. I also want to highlight Gurlaz Carr. She's working on another project I wanted to talk about, but didn't have time for today. But specifically, I wanted to mention Zach and Gurlaz because they're finishing up in May. So if any of you are on the lookout for a good postdoc, you can steal them from me. I don't have money for them at the moment, unfortunately, but they're great people, and they've developed a lot of good molecular biology skills in my lab. So they would be great for you. But other than that, I think I'll leave it off if there's no more time and open it up to questions. Yes.
Are there enough cattle in urban areas for the mosquitoes to not stop?
Are there enough? Oh, are there enough animals? Yeah. Well, that's a good question. I mean, it depends where you go. In India, definitely, there are a lot of cattle in the cities. I know that sounds a little bit weird, but they are kind of sacred animals, and they do kind of wander around the streets in a lot of places. So, yeah, in that case, that's possible. But you know, in some other urban areas, if they fed on something like rats or something, what's that?
Pigs.
Pigs, yes. So there are other options. Yeah. Other questions?
This is just about really great work. Thanks for coming. Great talk. I was interested in the expression of those odor receptors, you know, the topography of that, the extent to which they're clustered.
What do you mean with the topography? Like in other areas of the body, or?
Are they co-expressed in response to?
So I didn't talk about the receptors themselves. That's a little bit more complicated. I didn't want to. Yeah. There's a co-receptor is how they work. There's something called orco, and it plugs into all these other odorant receptors to make them work. But in the same neurons, there's only one odorant receptor of the specific types that are expressed typically. So, yeah, but there is some there's some interesting work that's been done on downregulating specific odorant receptors, and other ones will come up in kind of response to that. So yeah, it's kind of a complicated system.
Yeah, that's not always.
I tried to simplify it, but it's like, yeah, there's so many things going on. So, and, you know, and further from that, you know, there's just, you know, how are the insects wired? Where do these signals go? I mean, is it just because it has an association with human hosts doesn't mean that's attractive to them necessarily. It was a great study that looked at like expression very early on, like after the mosquitoes emerge as adults. They're not quite ready to blood feed at that point. It takes them about 4 days to really get everything stabilized, and there was high expression of this receptor that, well, it perceived a lot of human odorants, but they found out that was repellent. So they actually wouldn't host seek during that time, or were repelled from taking a blood meal just because they weren't quite ready to take a meal. So, yeah, there's a lot of cool stories I think out there. What else? Yeah.
How general the long width of the pathway is it? Like, would we just use them as sprays, or would we be giving every man and his dog diabetes?
Yeah, administering those kind of things is an open question, and that's also something that came up when I was working in Dr. Hill's lab. I mean, the characteristics that make a good drug don't necessarily make them good insecticides. So yeah, there would have to be some manipulations that are done, and I don't think you really want that anyway, right? Because yeah. Well, you know, we were working with, like, you know, antidepressants before, and it's like, oh, okay, well, let's put those, let's give everybody those, right? But, yeah. No, so yeah, there definitely have to be some modification, but it does give you kind of a chemical backbone to start working from. So, oh yeah, go ahead.
So all that insulin affect that.
How does insulin affect diapause? Yeah, that's something we're still unraveling. But you think about it, like during diapause, the insects have to induce this state of very limited nutrients. I mean, they're not getting any nutrient intake. So how are they going to manage, you know, blood sugars or whatnot? And there's been some look at that, but not very much. Also, whether or not the insect goes into diapause, there's some that have looked at that. You know, is it a reduction in the insulin signaling that induces diapause, or is it downstream of something else? So that's something we want to explore. But certainly the energy budget is going to be modified when they're trying to hunker down and survive the winter or the dry spell. Yeah.
So you have all these really interesting directions to go with the insulin peptides. Which one are you most excited about? Diapause or reproduction? Which one do you?
Well, anyone that works is good. No, it's I think diapause is interesting just because it's one that hasn't been explored. I mean, you know, it's kind of emphasized right there. There's a lot I think we could work out. I mean, I think, you know, reproduction is probably the next kind of low-hanging fruit that we could pick up, but exploring some of these other areas is interesting. I also think longevity is interesting. That's just involved in lifespan, and that goes across, animals, it appears to. I mean, even in mice, Drosophila, all these, and nematodes even. I mean, increased insulin signaling usually leads to a shorter lifespan. So that's kind of interesting. It's kind of a universal rule of life. But maybe some more things that are specific to Lygus. As I mentioned, there are some developmental effects. I want to explore that too. Because every once in a while in our colony, we have like these nice hefty insects, but they like these little tiny dwarf ones. And I'm like, why are you showing up? There's enough food for everybody. Why don't you have enough? Or what's limiting this? So yeah.
So I was interested in your strategy. Several graduate students here are learning how to do olfactometer bioassays, banging their heads. They're difficult, right?
Yeah. Yes, they are.
And so I was interested in your strategy for testing these human lo-fi and odorants.
Yeah.
How are you going to go about this?
We're going to do the Y-tube olfactometer. We've got specific odors, from our odor panel. I mean, that's very specific, but I think it's going to be a little bit more technically complicated because specifically with mosquitoes, they really need that carbon dioxide puff to get everything else going. A lot of times these odors aren't attractive in isolation, but in combination with carbon dioxide, suddenly they're attractive. So yeah, we may have to be working on blends of odors, and we've tried, we've put into proposals characterizing the odor profile of cattle, for instance. So can we see what's different about that and make a specific odor blend and pipe that down versus a more human-oriented odor blend? So yeah, it's not going to be easy because our early olfactometry assays have been just ridiculous. There's just so much error. I mean, mosquitoes just kind of do what they want, and we minimize that as much as possible, but getting them to behave is an issue. It's just replication is what it comes down to, I think. Yeah, providing them those conditions that they need is tricky.
Yeah, and blending is tough.
Yeah. So
sometimes it's better to start with full blend, start trying to subtract that out.
yeah, in the literature, I've seen a lot of versions of olfactometers too. I think people have kind of, thrown away their Y tubes just because it wasn't working. And, they do like single port, and it's like, are they attracted to just one odor, and they have different ways of scoring that, or, putting out two cups that have like, different odors in them and the mosquitoes land on them. So there's different ways of looking at it. I think it's, if you can get something to work, latch onto it. So, yeah, it is difficult. And the people that are successful at it, I mean, even if you look at their papers, it's like an N of 30 or something, or more, and the error bars are still pretty large. So it's challenging.
Just enough replication to get there.
Yeah. Stop the assay now.
Don't do another one.
Yeah. Any other questions?
One online.
Oh, okay. Yeah, sure.
Let me pull that up. You can read it.
I do want to.
You can probably get to it.
Yeah. Stop. Oh, yeah. Let's see. Oh, what was involved in inserting genes into the Drosophila empty neuron system? So this would be kind of your typical genetic modification. I think we used piggyback vectors for this. We sent it to a company to do it for us because, gosh, we're trying to do enough already recreating the wheel, and we just sent them a plasmid with the sequence on it, and they inserted it for us, and then we had to do a lot of breeding to get those mosquitoes, or not mosquitoes, Drosophila in the right genetic background. We lost a few lines, which I was not too happy about, Zach, but that happens. And COVID didn't help either with that. So we lost some stuff along there. I would have liked to present more receptor data, but it certainly set us back quite a bit. Good question. If there are no more questions, I do want to show you one more thing. So I am involved in a lot of other stuff. Whoops, a little too far. That I didn't get to talk about, but I just wanted to show you something cool because it's really more back to my roots as an entomologist, and it was super fun. About this time last year, I got a message from somebody at the wastewater treatment plant in town, and they were telling me about this fly infestation they had, and I was like, they were describing it to me, and I'm like, that doesn't sound like filth flies or drain flies or stuff. And what was happening was they have these kind of filtration devices to purify the water, right? And something was eating up the microbes that's supposed to purify the water. And they're like, we don't know what this fly is. And I was like, okay, send me this sample. And this is it. And it turns out it's a lemony crane fly, which is not something you find in nasty water. It should be pretty clean water. And looking through the literature, couldn't find anything, and eventually got in touch with this guy from the Smithsonian. He's like, oh yeah, this is a like incompletely described species. It's kind of mentioned in one paper from the 1930s, and that's about it. So there are a lot of cool things out there in entomology. It's just fun to keep exploring. So we're going to follow up on this in some way that we can. I'm probably going too many directions, but it was like a Friday afternoon, and I was like, I'm going to call it a day. And then I heard about this, and I just got super excited about it again. So there are a lot of fun things out there. All right. No more questions? Oh, yeah.
Like this life without
Yeah. You can see all this, there's all this algae and whatnot in these filtration things. This is like, they have all these big plastic blocks, it's kind of corrugated. This is kind of just pulled up, but they just spray the water over it continually. And this is obviously there's some cool microbe interactions in there, however they're digesting this. But they really seem to like that environment. And I guess it mimics whatever habitat they naturally inhabit. One thing that was interesting about this water plant was that the Truckee River system that goes through Reno goes from Lake Tahoe and on down to another lake, Pyramid Lake, and it's a completely closed system, so it never goes out to the ocean. So it's kind of this unique ecosystem, but it also flows into tribal lands, which is technically international water. So they have to have the water super clean before it leaves the wastewater plant. So this plant is a little different than most wastewater treatment plants. Kind of makes you rethink of what goes into the Wabash and other places. But anyway, so this is one of the last steps of their cleaning system. So I think it says that their water's super clean, which is good, but it allows these insects to thrive on it for whatever reason. So it'd be kind of cool to explore what sort of ecology they need, to survive. So hopefully we'll get to follow up on this in some way, but it's just kind of a fun thing. Yeah.
How would they even feel about, you can't like go and spray them because it's.
Yeah, you have to be really careful. And that's what I was trying to advise them on, earlier on. I was like, well, one of the things they can do is kind of flood these towers in some cases. Some of them are not stable enough to be able to do that. But that's one thing they tried initially. They were able to wash them out just by completely flooding them, because they can survive in this sort of trickling water environment, but they still need some exposure to air. But when they're completely flooded, they get killed off. But another thing was, I think they may have used one of the JH inhibitors like methoprene, for instance. Still not great for releasing into the river, but it breaks down quick enough. BT is another option. So, I was like, you want to avoid some of the neurotoxins for sure. Try to do stuff that's a little more benign. But those are my recommendations. I actually need to follow up with this guy. Kind of let this sit for a while, but it just keeps coming back to me. It looks like a fun thing to explore. It's kind of a guilty pleasure. This other stuff is better for getting out papers, but I don't know. It's fun. I'm a bug guy. I just love this stuff. All right. Any more questions?